Hi everyone, welcome to Bioinformatics Bytes, Episode 1. In this series, we'll break down big bioinformatic concept into bite-sized lessons. Today, we are talking about two file formats you'll bump into all the time in sequencing analysis, FASTA and FASTQ. If you have ever downloaded genome sequences or raw reads from NCBI or worked with next-generation sequencing, chances are you have seen these file formats. So let's dive into the details of these file formats. Okay, so a FASTA file is the most fundamental file format in bioinformatics. It is used to store biological sequences, either DNA, RNA, or proteins, in a very clean, readable format. Each entry in the FASTA file has two main parts. The first one is the header line, which always starts with a greater than symbol. This line gives information about the sequence, like its identifier, name, or description. Everything after the header is the actual sequence, written as a string of single-letter codes, A, T, G, C for nucleotides or amino acid letters for proteins. FASTA files are just plain text. They can be opened in Notepad or any editor. You'll often see them used when working with reference genome or conserved genes or protein databases. The file extension might be .fa, .fna or .faa, depending on whether it contains nucleotides or amino acids. And yes, one FASTA file can contain multiple sequences, each starting with its own header. Now let's take an example. So as you can see here, this FASTA file begins with the greater than symbol, which is the header, followed by some information about the sequence. Immediately after that, you will have the actual sequence. Now this FASTA file has multiple sequences, if you see, the first one, the second one, and each of them begins with its own header, that is greater than sign, some information about the sequence, and then the actual sequence. Notice one thing though, in the FASTA file you don't see any other information like quality scores or extra metadata, just bare essentials, the identifier and the sequence. So if you ever open a file that has extensions like .fa or .fna or .faa, you will see something like this in your file. Alrighty, so let's see what is a FastQ file. So FastQ file stores nucleotide sequence just like FASTA, but they also carry something extra, a quality score for each base in the sequence. This makes FastQ file extremely valuable when dealing with raw data from sequencing machine. Each read in FastQ file is made up of 4 lines per record. So it begins with @ symbol and contains a description. This could include information like instrument ID, run number, or read ID. The second line is the sequence itself. The third line is a plus sign, which is also called as a separator, and sometimes it might have the same description as in the header. And the fourth line, which is the most important one, is the quality scores. Now each quality score or each character in this line represents Phred quality score, which I will discuss about in our next slide. And it is encoded using ASCII, which is American Standard Code for Information Interchange. And they range from symbols like exclamation marks to J, which corresponds to Phred score 0 to 41. Again, we'll talk about it in our next slide. Now let's see one of our examples here. As you can see, the FastQ file, it starts from @ and then there is an information followed by the actual sequence, then a plus sign, which is a separator, and these are the quality scores, which are like hash, 8, B, C, C, F, G, which are encoded, as I mentioned, in ASCII format, and it often ranges from exclamation mark to J. Now, so if you want to summarize FastQ files again, they are sequence plus quality score, and I will talk about those quality score, also known as Phred score, in our next slide. And these FastQ files, they often come with an extension of .fastq or .fq, and they are commonly used as input for QC tools, aligners or assemblers in downstream analysis. Alright, so now that we have seen how FastQ files store both the sequence as well as some extra characters which are nothing but quality scores, and they are called as Phred scores. Let's see what they mean and understand one thing, that these quality scores are big deal in sequencing. Think of them as confidence rating from your sequencing machine. Each base call comes with a score that tells us how sure the machine is that it got that base right. Now, if you see this table here, the gray column corresponds to those quality score that you can see in the FastQ file, and they are stored using ASCII character, which ranges from exclamation mark to J, and they correspond to the Phred score, which ranges from 0 to 41. Now, if I say a Phred score of 20, what does it mean? So that could be calculated using this formula here. So let's say I substitute Q as 20, which is our Phred score. It means that there is one wrong base in every 1000 call, which means that machine has 99% confidence for that particular read. Now let's say a Phred score of 30. Again, if I substitute 30 here, now we are talking about 1 in 1000 chance of error. And if we have higher score, let's say we have a higher score of 40, a Phred score of 40, even better, which means that our error rate drops to just 1 in 10,000. Now let's take an example to simplify this. Let's say you are sequencing a human genome at 50x coverage, which means you'll generate around 150 billion base calls. In every base, if every base had a Phred score of 20, that's 1% error rate, right? What you would expect, which means that if you substitute this formula again, you would expect 1.5 billion potential errors, and that's a lot of mistakes, that's a lot of noise to manage, right? But if your reads average a Phred score of 30, which is 0.1% error, the number of potential mistakes drops to just 150. It's not just, but it drops to 150 million errors, which is 10x improvement in readability. So what do you understand by this is, the higher the Phred score, lower error probability. The Phred score helps you filter, trim and trust your sequencing data. And remember that they are essential for accurate results in anything from variant calling to genome assembly. So the only thing that I want you to remember in this slide is, higher the Phred score, lower error probability. And usually the Phred score of about 33 is considered to be good, again in specific applications. Alright, so to summarize, FASTA is your basic format. It stores just the sequence, whether DNA, RNA or protein, in a simple human readable format. Each entry starts with a header marked by a greater than symbol, followed by the sequence itself. You'll find FASTA files everywhere, from reference genome to protein databases. Whereas on the other hand, FastQ is designed for raw sequencing data. It builds on FASTA format but adds one crucial element, and that is the quality scores. Okay, here they are. Each read in FastQ file includes 4 lines, one for the header, one for the sequence, a plus sign which is also called a separator, and one for quality string. These quality scores, encoded as ASCII characters, tell you how confident the sequencer is in each base call. So how do they compare? FASTA is lightweight, great for storing clean reference data, whereas FastQ is more detailed, essential when you're analyzing raw reads and need to access quality. So in short, FASTA can be used for alignment references and databases, whereas FastQ when working with raw sequencing read and quality control. Okay, now that we know what FASTA and FastQ are, let's see where they actually show up in real pipeline. Also, let's see some of the tools that we'll use in that pipeline. So we always start with FastQ reads, that's raw data straight from the sequencer. The first stage is QC, the most important step, where you'll use tools like FastQC, which takes in FastQ file and spits out a report. You can use a next tool like MultiQC, which bundles all those reports into one dashboard. The next stage is trimming or cleaning. You can use tools like Trimmomatic, CutAdapt or FastP, which takes in FastQ file and spits out FastQ file again. They basically trim adapters or low quality bases, keeping only the good stuff or good sequences, okay? The next step is file wrangling, optional. If you need to convert or sample or filter, you can use something called as SecTK or CKit, which handles both FASTA and FastQ file formats. They can turn your FastQ into FASTA, grab a subset of read or check lengths. Another thing called as FastQ validator, again an important one, which focuses only on FastQ, making sure formatting won't crash your pipeline. The next stage is alignment and mapping. So here you use this classic combo of FastQ read, which is aligned to FASTA reference file. So for that, you'll use tools like BWA or Bowtie2 for DNA short reads, STAR or HiSat2 for RNA-seq, Minimap2 for long reads or mixed cases. The outputs are SAM or BAM files. Again, we'll talk about these two files in some of our next videos. For post-alignment tools, could be SAMtools, BCF tools, which work on SAM, BAM or VCF files for sorting, indexing and variant calling. The next step is compression and indexing. Compression, which can help, which can keep files manageable. You can use gzip or bgzip to compress anything text-based, and other tools as well to manage those files, right? The next one is workflow, again an important one. You can use something that can glue everything together so that they are reproducible as well, and for that you can use SnakeMake, Nextflow, very famous one Nextflow, or WDL. They orchestrate every step from FastQ input to VCF output, so reruns are painless. Rule of thumb, FastQ, again it's reads for raw and raw reads with quality score, and FASTA reference or clean sequences, okay, with just a header and the sequence. So that's all for the tools section. Okay. All right, so quick recap before we close. We looked at what FASTA file is, what a FastQ file is, and how Phred scores tell us the confidence in each base call. We also walked through the essential tools you'll use in a typical pipeline, from QC and trimming to alignment and post-processing. That's it for this episode of Bioinformatic Bytes. If you are into these bite-sized breakdowns, hit like, drop a comment and subscribe so you don't miss the next one. In episode 2, we'll keep unpacking big bioinformatic concept into small, easy chunks. Thank you so much for watching. See you next time. Bye.